Okay, so in previous lecture 2, we studied about Mendelian inheritance. So in Mendelian inheritance, most of the experimental results, they were in the ratio of 3:1 if it was a monohybrid cross, or it is 9:3:1 for dihybrid cross. So there was a set outcome, right? And this was due to the reason that all traits which Mendel studied or which Mendel considered, it was controlled by, traits were controlled by single gene. So this is not the case for all plants, for all experimental crosses. So there was certain phenomenon which do not follow the Mendel's ratio. So there was deviation from these ratios. So those phenomenons are non-Mendelian inheritance. Those phenomenon shows non-Mendelian inheritance. So we'll take up all those phenomenon. The first phenomenon is incomplete dominance. So the incomplete dominance is, there are red flower, there are white flower. The red flower is controlled by, the red trait is controlled by homozygous dominant R allele. White trait is controlled by small r allele, the recessive allele. So in F1 generation, the expected outcome was a hybrid which will produce red flowers. But this was not the case. Here, the hybrid produced a different kind of phenotype, which was pink flower. Right? So it was a pink flower. When selfing was done in F1 generation, so the F2 phenotypes were red flower, pink flower, and white flower. So the ratio, the phenotypic ratio in this case was 1:2:1. One was red, 2 are pink, and 1 is white. But the expected outcome was 3:1. So the scientists, they expected the 3 will be red, 1 will be the white. But this was not the case. So this was the case of incomplete dominance. The dominant allele, it expressed, there's no doubt, it expressed, but it do not expressed completely. It do not express completely. It produced a pigment which was not sufficient enough to produce the red color. Right? So this is incomplete dominance. It has shown dominance, but that dominance was not complete. Right? So in this case, the genotypic ratio is 1:2:1. So there was 1 homozygous dominant, 2 hybrids, 1 homozygous recessive. So genotypic ratio and phenotypic ratio in case of incomplete dominance is same. So the second phenomena which do not follows the Mendel's inheritance is the lethal genes. This is a bit confusing. So you can change the genotype here. So scientists took 2 mice, yellow colored mice, which were hybrid, right? So these are the genotypes of these 2 yellow mice, right? So scientists made a cross between 2 yellow mice and both of them were hybrid, right? So the possibility of next generation, the genotypes of next generation will be let me complete this table. So the possibility of the genotypes in the next generation, it will be YY, then hybrids, and then recessive, homozygous recessive in the ratio of 1:2:1. So this was the expected genotypic ratio, as well as the expected phenotypic ratio was 3:1. This was expected phenotypic ratio. Fine, but what happened in this case? The mice which inherited the two homozygous dominant alleles, they died. So dominant allele in homozygous condition was lethal. It caused the death of the organism. So the outcome of this cross was the mice were found in a ratio of 2:1 instead of ratio of 1:2:1. And the phenotypic ratio was, again, the phenotypic ratio will be, these two will appear as yellow. Two mice were yellow and one was brown, which is again the phenotypic ratio is different from the phenotypic ratio of the Mendel. So the next type of gene interaction is the complementary gene interaction, where the Mendel's dihybrid ratio 9:3:3:1 is modified to 9:7, right? We will see how. So all these interactions which we are going to study in the next topic are non-interallelic gene interactions. So that means we'll see how two non-alleles, they interact together and they affect the phenotype. So in complementary gene interaction, two dominant alleles, allele C and allele P, these two dominant alleles, they are non-allelic. They do not show any phenotype. They produce white color flowers. But when these two alleles, non-allelic dominant alleles, they come together, they show a phenotype, right? So when both the dominant alleles are together, they produce purple color, right? So in this case, dominant allele C doesn't produce any phenotype. It gives white color flowers. Again, dominant P produces white color flowers. But when these two dominant alleles are together, they produce purple color flower. Upon self-fertilization, it will produce all these possible offsprings, right? So all these offsprings where dominant C is with dominant P, it produces purple colored flowers, right? So we'll take the example of this. This offspring will have white color flowers because there is no dominant C along with dominant P. Similarly, in this case, this will also produce white flowers because there is no dominant P along with dominant C. So next type of gene interaction is the supplementary gene interaction. So this is similar to complementary gene interaction in a way that again this is non-allelic gene interaction, or you can also call it as interallelic. So in this case again we will see how two different alleles, they will interact together and affect the phenotype. So in case of complementary gene interaction, there were two dominant alleles which were having no phenotype, but when they were together, when these two dominant alleles were together, they were producing a phenotype which was purple color flowers, right? Now in this case, in supplementary gene interaction, you'll see there are two dominant alleles. C has a phenotype. It produces black color in mouse. Right? A has no phenotype. A produces no phenotype. But when these two dominant alleles, non-alleles basically, they come together, they produces a different phenotype. That was an agouti mice. Agouti is dull brown. Right? So in this case, you can see when two agouti mice, they were crossed together. As why they are agouti? Because they have two dominant allele here, right? So when they are crossed together, they will produce three kinds of mice: agouti, 9 agouti, 3 black and 4 albino. So we'll take one by one. Why this is agouti? Because it has both the dominant alleles, right? Why this is black? Because it has only one dominant allele, that is C, which is responsible for the production of the black phenotype. And why this is albino, white? Because it has a dominant allele which alone do not produce any phenotype. So in case of supplementary gene interaction, the ratio 9:3:3:3:1 is modified to 9:3:4. So the next type of gene interaction is the inhibitory gene interaction, where the ratio is modified to 13:3. Again, this is a non-allelic kind of gene interaction, right? So in this, we'll take example of rice color. Rice plant, we'll take the example of leaf color in rice plants. So this dominant allele, P, it produces purple leaf color. This is responsible for purple leaf color. Whereas the recessive allele is responsible for the green leaf color. But as I have already told you that it's a kind of non-allelic gene interaction, but these two are the alleles of same gene. But there is another allele, I. We'll see how this allele, this non-allelic gene interacts with this dominant allele. This allele, this dominant allele, whenever is present along with this dominant allele, this will inhibit the color produced by dominant P allele. Otherwise, dominant P allele is responsible for purple color. But when this dominant P allele is present with another allele, dominant I, it will produce again green color. So in this kind of interaction, I is inhibiting the effect of P, and both I and P, they are non-allelic genes. Right? So here across, this rice plant has green colored leaves because P is in recessive state. This is purple colored leaves because P is in dominant state. Now mating was done, hybrid was produced. So this is green because there is a dominant I present along with dominant P. So I has inhibited the color, the effect of P. Right? So when selfing was done, these were possible offsprings. So this is green because dominant I is present with dominant P allele. Right? We'll see why this is green. This individual is green. This is green because again I is present with dominant P. This is purple because there is one dominant allele present here which will cause the color of the leaves to be purple, right? This is green because in recessive state These alleles are responsible for green leaves, right? So this is inhibitory gene interaction where Mendel's 9:3:3:1, which is a typical ratio, is changed to or modified into 13:3. The next kind of gene interaction is the duplicate gene interaction. Here two alleles, two dominant alleles of different genes, they are producing same kind of phenotype. Same kind of phenotype. So here the example is of Capsula plant. Capsula plant has two types of seed capsule. Seed capsule is the seed covering. So the two types of seed covering are oval shaped or triangular shaped. So this triangular shaped seed cover, so these are the seeds and this is the seed capsule. It is controlled by two different dominant alleles, right? So A is also producing this type of phenotype, B is also producing this type of phenotype. An oval shaped seed capsule is possible only when the alleles are in homozygous recessive condition. Alright, so this is duplicate gene interaction. So if you do a cross between two plants, one with triangular seed coat, other with oval shaped seed coat, so they produced a hybrid which was having a triangular seed capsule. So when you will do selfing in this, the outcome will be, right? So this is the outcome in the form of Punnett square. So you will see the two dominant alleles, they were producing same kind of phenotype. Again, this one together producing same kind of phenotype. All are triangular except for this, right? So only the alleles in recessive condition, homozygous recessive condition will produce oval shape seed capsule. We see another genotype, consider this. So this has a dominant allele, B, right? So that's why it is producing triangular seed coat. If you consider this, it has A, dominant allele, right? So this is producing again triangular seed capsule. So this is duplicate gene interaction where the phenotypic ratio is modified to 15:1. Okay, so next kind of non-allelic gene interaction is the polymeric gene interaction, right? So in this polymeric gene interaction, you'll see that there are two dominant alleles, non-allelic dominant alleles. Both of them, they produce same phenotype. Both produce same phenotype, but when they are together, they enhance the phenotype. They intensify it. So we'll understand this with an example of on length on barley fruit, right? So what are on are these hair-like extensions, present on the barley fruit. So these awns, they help in the protection of the seeds from the animals, and also they help in dispersal of the seeds. So we'll take the example of on length on barley fruit. So in this genotype AABB, they will produce long on because A and B are together. Also, this genotype will be responsible to produce long on. Again, dominant alleles are together. This genotype will produce a barley fruit without on. So this will be onless. And this barley, this genotype will produce medium length on. Reason being because dominant allele is, there's a single dominant allele. Similarly, this genotype again will produce medium length on. So the point is, alone they produce some phenotype, alone we also produce some phenotype which is medium length on, but together they intensify the phenotype, they produce long length ons on barley fruit. So you can look at this cross, this is long on, this is onless variety, both were crossed, and there was a hybrid produced in F1 generation. So very obviously both the dominant alleles are together, so it will produce long on. So when selfing was done, the plants, they were found to be in a ratio of 9:6:1. 9 were having long on, 6 were having medium on, and 1 was showing to be onless. So this is how, due to this polymeric gene interaction, the ratio was modified to 9:6:1, which was expected to be 9:3:3:1. So now we will practice some question on non-Mendelian inheritance. So here they are asking that in Lathyrus, so Lathyrus is the sweet pea, the cross between two purple flowered plants, they gave a white flowered progeny. So we discussed about the purple colored flowers in sweet pea in case of complementary gene interaction, where this genotype and a plant with this genotype both had produced white colored flowers. So you can say they were producing no phenotype, but when a hybrid was produced in F1 generation, it has shown purple colored flower. So purple colored flowers, right? So this thing was possible because of complementary gene interaction. The two dominant non-allelic dominant genes together, they had produced a new phenotype. But now the question is, when two purple colored flowers, when these two flowers, they were mated together, a white flowered progeny was produced. Now, how is this possible? How purple colored flower can give rise to white colored flower? So this is because the gametes, during gamete formation, alleles, whether allelic or non-allelic, they segregate independent of each other. So they follow the rule of segregation, right? Chromosomes or genes or alleles, they follow the rule of segregation during cell division. So this is possible due to this phenomena. So the answer is segregation. Second question is, the two non-allelic genes, they produce new phenotype when present together, but fail to do so independently. So again, you can take the example of sweet pea, where they were producing white flower. Alone they were producing white flower. But the hybrid, which had both the dominant alleles, non-allelic dominant alleles, so it had produced a different phenotype, new phenotype, which was purple flowers. Now this is due to complementary gene interaction, right? Third question is, a monohybrid cross between two plants, one having 24 centimeter long leaves and other is having 12 centimeter long leaves. But the F1 hybrid all are having 18 centimeter long leaves. So this is clearly this is not the case of multiple allelism, complete dominance, recessive dominance. This is a case of incomplete dominance, right? Flower color in Mirabilis jalpa. This is Japanese 4 o'clock. This plant is also known as Japanese 4 o'clock, is an example of So Japanese 4 o'clock plant, it had red color flowers. When crossed with white colored flowers, it produced pink flowers. So it's the case of, this was the case of incomplete dominance. But you know this very well, that incomplete dominance do not follow the pattern of Mendelian inheritance. So it's also non-Mendelian inheritance, right? So the answer is D, option A and B. Now, next question is the expected phenotypic and genotypic ratio in case of incomplete dominance in Mirabilis R. So in incomplete dominance, the ratio was the hybrid, the pink flowers, when they were self-pollinated. So they produced what? I'm completing this table. They produced 1 red, 2 pink and 1 white colored flowers, right? So the phenotypic ratio was 1:2:1, and similarly the genotypic ratio is 1:2:1. So both the ratios are same in case of incomplete dominance. So the answer is 1:2:1 and 1:2:1. Then, so the next question is question number 6. A blue Andalusian fowl is the product of mating black fowl. So that means this is the black fowl. It is mated with white fowl, so it has produced a blue fowl. And a cross of blue fowl with another blue fowl, so this is the case of incomplete dominance. So it will produce something in a ratio of 1:2:1. We know that. So 1 will be the black, 2 will be the blue, and 1 will be the white fowl. So the answer is 1 black, 2 blue, 1 white. So answer is B. Second, the same phenotypic and genotypic ratio is seen in incomplete dominance. Then question 8, the gene interaction in which either of dominant gene is enough to give rise to same phenotypic effect. So that means there's a dominant allele which produces some effect. The another dominant allele, non-allelic interaction, it's about non-allelic interaction. The another dominant allele is also producing the same effect, right? And they are enough to give rise to the same phenotype, right? So this is the case of duplicate genes. Together also they produce same effect. They do not show any additive effect, right? They produce same effect. If A is producing the 74 centimeter long leaves, B is producing 74 centimeter long leaves, A and B together are also producing 74 centimeter long leaves. So this is the case of duplicate gene interaction. The ratio of dihybrid cross is modified to 9:7 in. So this happens in supplementary gene interaction. The ratio is modified to 9:7, right? Then the next question is, among following, which is the intra-allelic gene interaction? Intra-allelic means these two alleles, the interaction between these two alleles, or these two alleles. They are similar alleles. They are the alleles of same gene, right? So the interaction between these alleles will be known as intra-allelic gene interaction. So this is in the case of incomplete dominance. As you have seen, the hybrid produces pink color. So this is because how R is interacting with this R. Dominant allele is interacting with recessive allele, right? Next question, among following which are the interallelic gene interaction? That means how this allele is interacting with some other allele, or this allele is interacting with some other allele, and both are not the same genes, both are not the alleles of same genes, right? So this happens in supplementary, complementary, duplicate. So this happens in E, A, B, C, all the three. Incomplete dominance shows intra-allelic gene interaction. Right? So next question is, which of the following is also known as blending inheritance? Blending means where the character is blended, mixed. So this happens in case of incomplete dominance. Right? So incomplete dominance is also known as blending inheritance.